Assalamualaikum warahmatullahi wabarakatuh. Shalom, Om Swastiastu, Namo Buddhaya, Salam Kebajikan. Good morning to all of us, and welcome back to Day 3 of the virtual course 2024, organized by the Faculty of Pharmacy, Universitas Indonesia. Allow me to introduce myself. I'm Kirana Dinda Sakina, an undergraduate student of Faculty of Pharmacy, Universitas Indonesia. Ladies and gentlemen, allow me to read out our agenda for today. Today's course will be started off by Lecture 5, titled Exploring the Therapeutic Potential of Natural Products Through In Vitro Bioactivity Studies, which will be delivered by Dr. Do Bích Hằng from Ton Duc Thang University, Vietnam. Followed by Lecture 6, titled In Vivo Investigations of Natural Compounds in Disease Models by Dr. Za Bu Chan from Ton Duc Thang University, Vietnam. Next, agenda is Lecture 7, titled Boron Neutron Capture Therapy, its application for targeted cancer therapy, delivered by Professor Eiji Matsura, who is a professor of Okayama University, Japan, and also an adjunct professor at Faculty of Pharmacy, Universitas Indonesia. Then, we will proceed with the break session. After the break session, we will continue with Lecture 8, titled The Potency of Bottle Brush Polymers as Drug Delivery Carrier by Dr. Ernie Sagita from Universitas Indonesia. Followed by Lecture 9, titled Lipid Nanoparticles for mRNA Delivery and Strategy for Tissue-Specific Targeting by Dr. Zihni Ahda Islami Mazrat from Monash Institute of Pharmaceutical Science, Australia. And lastly, we'll be hearing a closing remark from the Dean of Faculty of Pharmacy, Universitas Indonesia, Professor Arya Nuas. Ladies and gentlemen, we are now ready to begin the first and second lecture of the day. The first lecture is entitled Exploring the Therapeutic Potential of Natural Products Through In Vitro Bioactivity Studies by Dr. Hang Bích Do from Ton Duc Thang University, Vietnam. The second lecture is entitled In Vivo Investigations of Natural Compounds in Disease Models by Dr. Za Bu Chan from Ton Duc Thang University, Vietnam. Before the lecture starts, it is my pleasure to introduce our moderator for this session, Ms. Kalista Andini Mulyadi from Universitas Indonesia. Ms. Kalista completed her Bachelor of Pharmacy and Professional Pharmacist degree at the Faculty of Pharmacy, Universitas Indonesia in 2018 and 2019, respectively. She obtained her Master of Pharmacy degree at the Faculty of Pharmacy, Universitas Indonesia in 2023, specializing in pharmaceutical chemistry. She has experience as a community pharmacist at Apotek FIBA in 2019 to 2022. Currently, she is a lecturer and researcher at Faculty of Pharmacy, Universitas Indonesia in the Laboratory of Pharmaceutical Medicinal Chemistry and Bioanalysis with research in analytical method development and bioanalysis. Without further ado, Ms. Kalista, the floor is yours.
Thank you, MC, for the introduction. Good morning, everyone.
Good morning.
Good morning.
My name is Kalista. Be presented by University Big Edo State. Dr. Do Bích Hằng is a lecturer in the Faculty of Pharmacy. She gained her PhD of Medical Science from University of Ulsan, South Korea on 20. 2017, published in journal Purdue. Please welcome the Dr. are yours.
Hello. Hello, Calista. Miss Calista, can you hear me clearly?
Yes, I can hear you clearly.
Okay, thank you. Thank you so much for introducing me. Okay, so good morning, lecturers and students from Universitas Indonesia. My name is Bích Hằng. Just call me Hằng. So now I will share the screen. Wait a moment. Maybe I should say another one. Oops. Can you see my sharing now?
Yes, I can see your screen.
Okay, so may I start it now?
Okay, please.
Okay, thank you. Okay, good morning, everyone. Um, so sorry, something wrong with here. Okay, previously, you heard Dr. Dong. He introduced about how to extract or isolate the compound from natural products. So now I will keep going on with that project. That means when you have the compound or the extracts, then you need to test whether that compound or extract have any activity or not. And firstly, we're going to test the in vitro activity. So that's what I wanted to introduce today. Maybe I do not need to introduce myself anymore. I introduced already. Actually, my research only focused, previously I focused on recombinant protein, how to express or produce recombinant protein, mostly in E. coli, in yeast, or in mammalian cells. But recently, I moved to test the bioactivity of natural products. For example, antimicrobial, antioxidant, anti-inflammatory, or anti-cancer. Just some screening on some bioactivity of natural products. As you know that there's so many drugs come from natural products. For example, paclitaxel, penicillin, or some other. They also isolate from nature. So you mean nature is a really potential source for drugs. And when you isolate the compound or extract, you need to test the preclinical first, and then you move to clinical trials. Maybe students, you know about this already, so it just go a little bit about this. So in the preclinical, we have the in vitro first, and then in vivo, and then we go to 4 phases of clinical trials. That means we test on human. And I want to go to some methods of bioassays. First, in silico, next in vitro, ex vivo, in vivo, and human or clinical trials. Anybody know? Some student may ask student, do you know what is in silico? In silico study? Oops. Wait for me a moment. Oops. If you have the answer, just type in the chat box or just raise your hand, please. Do you know what is in silico or in vitro, in vivo, ex vivo? So please tell me and share with everybody. You still hear me, right? Hello.
Dr. Hang,
Yes.
if if maybe about in silico, you use computational
Hello.
method for
Okay.
bioassay.
Hello.
Okay. Hello.
Oh,
Umara,
Hello.
right? Yeah.
Hello. Good morning.
Yeah, good morning.
I want to give my view on the in silico studies.
Yes, thank you. So please share with
Uh,
everybody.
I know it as the use of computer-based methods and stimulation to study biological and chemical systems, particularly for drug discovery and development. Then for in vitro studies, there are experiments conducted outside the living organism, particularly in the laboratory, and they are done in the test tube. Then for in vivo studies, this one is done on animals like the mice, the rats, and so on. Yeah, that is what I can say. Then for clinical trials, this one is now done on humans.
Yes. Okay,
Yes,
thank you
thank
so
you.
much. Thank you so much for your answer. Yes, really, it's correct. Yeah, correctly. In silico, that means, as he said, that in silico, that means we use the computer screening to screen the whether that compound has activity or not. For first, from a lot of compound, we need to screen first. So we use the computer, the software, to test whether that compound has activity or not. And then we go to in vitro, that means we test on the cells or in the enzymes outside the body. And ex vivo, that means we test on the tissue outside the bodies. And then in vivo, that means we test on the inside the body in the animals. And then human trial test on the human. Yeah, correctly. Thank you. And then let's go next. Oops. And there's so many in vitro activity, but because the limitation of the time today, so I just introduced some that I work on it. That is antimicrobial, antioxidant, and anti-cancer assays. Firstly, I want to go to antioxidant assays. First, we need to talk about free radicals. Maybe you know all about free radicals. There's a lot around us. That is the O2, OH, H2O2, NO, O3, and so on, some other. That means they can form from UV light, air pollution, and there's radiation, smoking, and some, a lot. There's so many reasons forming free radicals around us. And the free radical, if the free radical can attack to the cells by damage the DNA, damage the cell, lead to the cell's death. When the cell death, it will affect to our body, and it can cause a lot of disease. For example, aging, some inflammation, some cancer, and some other disease. And to reduce the harmful of the free radical, we use antioxidants, right? Antioxidant is a compound that will give the electron to the unpaired electron of the free radical, so it will neutralize the free radical, so it will not more active, so active anymore. So it will protect our body. And there's a lot of sources of antioxidants, mostly can be from fruits, vegetables, yeah, and some functional medicines. Yeah, there's a lot of antioxidants. And to test the whether a compound has antioxidant activity or not, we have some in vitro activity, some in vitro assays. For example, ORAC, CORAC, QPROC, DPPH, FRAC, PFRAC, APTS, TRAP, and some other. They're mostly there's the same mechanism. That means we're gonna use the compound that has the function is the free radical, and then we're gonna add our compound. If our compound has the antioxidant activity, our compound will give one electron to the free radical, so it will change the color, and then we will measure the color change to know that whether our compound has activity or not. And now I just want to focus on the TPPH that is simple. That is simple and standardized already. Many paper and many people use this method. About TPPH, TPPH has the role as the free radical. TPPH lack of one electron. So when we dissolve TPPH in the solvent such as methanol, it will have the color yellow. If the antioxidant give one electron to TPPH, the color will change to the purple. So we will measure the purple color. The spectrophotometer normally we use at 500 for the to detect the purple color of TPPH. And firstly, we prepare the sample, we prepare the TPPH, and then we mix the sample and the TPPH. Normally we mix in the dark because the light can affect the activity of the sample. And then incubate it around 30 minutes, and then we measure using the spectrophotometer at 517 nanometer. And we often prepare several different concentrations of the sample, so we can make a graph or a line and then we can measure the IC50. When we have the result, we can calculate the percentage of inhibition by using the absorbance of the control. The control, that means without our sample. And then minus the absorbance of our sample, divide to absorbance of the control, and multiple of 100%. Using different concentration of the sample, we can make a graph like this. Normally, we often use one standard. Normally, I use ascorbic acid because the positive control, because ascorbic acid is a really popular compound, really high antioxidant activity. In here, I test different extract of the Garcinia group. This is one kind of plant quite popular in Vietnam. And then we extract using different kind of solvent, for example, ethyl acetate, methanol, hexane, chloroform. Yes, and then we compare the antioxidant activity of different extracts. From the graph, we can measure the IC50. Normally, I use the software, for example, GraphPath, to give us the IC50. So, and then we will compare the IC50 between different samples. If the IC50 is smaller, that means the activity is higher, right? So we try to figure out the samples with low IC50. That is the really basic technique to measure to screen the antioxidant of the extract of compounds. And then I will move to antimicrobial. Antimicrobial, that means the compound extract can inhibit and can kill the bacteria, right? In here, I have the figure of two kinds of bacteria. Any students, so please tell me which one, what kind of bacteria is this? What kind of group of bacteria in the left side and the right side? Anybody? So you can write down in the chat box. Raise your hands, please. And the lower figure is the figure of gram staining.
Hello.
Hello.
I think the one on my right.
Yes.
The one, yeah,
you're right.
I think it is E. coli.
Yes.
Then the one, this one, the one on my left is the Staph aureus.
Staphylococcus? You mean what? On the left is?
Coccus aureus.
Coccus. Okay, thank you. So actually, it's just meaning that, yeah, thank you for your answer. I just want to mean that the right side is the gram-negative. Yeah, E. coli is a gram-negative bacteria. Here, you know that it is gram-negative. So gram-negative bacteria contain two membrane, the outer membrane and the inner membrane. Between them is the periplasmic space. And for the gram-positive, on the left side is the gram-positive. The gram-positive bacteria only have one membrane, but outside is a very thick peptidoglycan. For the gram-negative, really thin peptidoglycan. And for lower one is the gram staining. If the gram staining is the purple, blue, purple, that means the gram positive. And the color is some kind of pink. It is the gram negative. Yes, you know that bacteria cause a lot of disease from quite simple, for example, cough or sneeze, to really some serious disease, for example, pneumonia, diphtheria, tuberculosis, anthrax, tetanus, or something. And the bacteria are always around us every day. And maybe you know this, it is the to kill the bacteria. Normally we use the antibiotic, and the problem now, not only in our country but in the world, that is the problem of the antibiotic resistance. That means the bacteria are really smart. They can against the antibiotic. Often, so the researchers always try to find out the compounds or the extract to help, just to help or support the antibiotics to kill bacteria. Can wait for me a moment. Okay, can we continue? Yes. There's some method to, yeah, sorry. Some method analyze the antimicrobial activity. For example, we can use the diffusion methods. That means we take the whether when our compound is fused into the plate, we can see whether our compound can kill the bacteria or not. Or we can use the thin layer chromatography, TLC. TLC, that means we can separate the compounds into the TLC, and then we apply TLC onto the bacteria to see that the compound on the TLC can kill the bacteria or not. We can use the dilution method. Dilution, that means we dilute our sample to different concentrations, and then we add the bacteria to see which concentration can kill the bacteria. Or time kill test, that means we will measure at different times, for example, 2 hours, 4, 6, 8, 12, or 24 hours, and we can see at which time our compound can kill the bacteria. Or we can use the ATP bioluminescence assay. That means we can measure the ATP, that means the energy from the bacteria, by using the bioluminescence. Then we measure the bioluminescence. We can know how many cells still alive. Or we can use the flow cytometry. Using flow cytometry, we can measure the cells alive after treating with the sample. So here I will introduce two quite simple and often used to screen the activity of the compound. That is the diffusion method, number 1, and number 3 are dilution methods. For the diffusion method, we often use on the agar blade. Agar blade can be TSA or the MHA, the blade to make the blade. Firstly, we prepare the blade, and we prepare the samples. Normally, samples we can dissolve in DMSO or any solvent that can dissolve our sample. Normally, I use DMSO, and then we prepare the bacteria. Which will prepare any bacteria you wanna test. The bacteria strain you can get from ATCC, and we culture the bacteria, and then we will measure the amount of the bacteria. Normally, we get the amount of the bacteria is around 10 to the power of 8 CFU per ml, because at this amount is enough for the cells to thread on the plate for this kind of test. Or we can use the Max Planck. Yes. When we prepare the bacteria at the suitable concentration, and then we're gonna thread. Okay, I don't have the At first we're gonna thread the bacteria on the plate. You can use the cotton swab, the sterile cotton swab to thread the bacteria on the plate. When you thread the bacteria on the plate already, then you can make some hole, or you can use some paper, sterile paper. That means sterile paper with the compound already. Normally we use the sample, the positive control and the negative control. The positive control is the antibiotic, and the negative control is the solvent that you dissolve the compound. Yes, you can dip the paper into the samples and then add directly onto the plate. And then we can incubate from 24 to 48 hours at 37 degrees in incubator. And then for the next day, we can measure the diameter of the inhibited zone. So the inhibition zone, that is the zone without the bacteria living. Yes, you can see the zone here. If our compound has no zone, that means no activity. If the inhibition zone the bigger, that means the activity is higher. And oops, I'm sorry, I like up slide. I just remember I still have one slide about introduce about dilution method, but somehow I see deleted here. So another method is the dilution. So from dilution, you can make some different concentration of the compounds, and then you add the bacteria into each concentration. Also incubate 24 hours. After 24 hours, you can see if the which concentration the cells appear, and which concentration you cannot see any bacteria living. And that one from that one you can measure the MIC. The MIC, that means the lowest concentration of the agent that inhibits the growth of the cells. Yes, that is the
Pardon
MIC.
on the method of dilution.
Yes, sorry.
Come again on the dilution method. Like you said, you measure the different concentration of the sample, then you
Yes.
introduce the bacteria. Yeah.
Yes, and then I add the bacteria, and then I incubate at 37 degrees for 24 hours. And then for the next day, I will check at which concentration has the bacteria and which concentration has no bacteria living growth. And at which concentration we cannot see any bacteria, that means the MIC, that is the lowest concentration. There's no bacterial growth. And I'm sorry because we have no slide here, so just imagine about that. Yes, and from the MIC, the tube without the bacterial living, we can thread. We take a little to thread onto the plate and incubate and continue incubate for 24 hours. And if the plate we cannot see any colony, that means the MBC. MBC is the lowest concentration that kills on the organism. So on the plate, we cannot see any colony. We still can see some colonies on that. That is the MIC. Is it okay? Can we move to another one? Okay. So the final one is the anti-cancer. For the anti-cancer, we know that the cancer, there's so many kinds of cancers. Yes, and once cancer metastasis happen, it's really difficult for the patient to be recovered. And I don't know in your country, but in my country, normally, when the patient with the cancer, with a tumor, normally, firstly, we remove the tumor first, and then we treat the patient with radiotherapy or chemotherapy or combine them together to remove the remaining cancer cells inside the body. But as you know that chemotherapy is really toxic to the patient. Normally, it can not only kill the normal cells, not only kill the cancer cells, but also the normal cells. And then the patient has to sustain a really, difficult for them to recover from treatment of chemotherapy. So therefore, a researcher try to find out the extracts or the compounds from natural products we can add together with the chemotherapy to reduce the side effects for the patient or to increase the activity of the chemotherapy. And first, once we get the compound or the extract, we need to test whether that compound extract can kill the cancer cells or not. So that is the cytotoxicity activity. Normally we can, there's a lot that is MTT, SRP, WSC1, and so on. And now I introduce MTT, that is the simple one. For the MTT assay, we're gonna measure the, we know that the cells has the, this is the mitochondria. Mitochondria can do the cellular respiration. When the cells do the respiration, they will produce the NADH and NADPH. So from this one, that is the dehydrogenase, the activity of the dehydrogenase. When we add the MTT, if our cells are alive, the cells has the dehydrogenase will change MTT to MTT formazine. MTT formazines with the color is purple, really beautiful purple. So then we just dilute, just dissolve the purple formazine, and we measure the purple color of the MTT formazine. If our sample is more purple, that means the more cells are still alive. That means that our compound has the low activity. Here is the procedure of the MTT assays. Normally we use the 96 well plate. At first we seed the cancer cells and the cells into the plate first. Normally we add around 10 to the power of 4 or 10 to the power of 5 cells per well. It depends on the growth of our cells, depend on each cells. And then we incubate around for 24 hours for the cells to grow, and then growth stable, for the cells to stable. And then we move to, and then we gonna add the sample. Can our compound or our extracts. We often add together with the positive control, and negative control together. And then incubate for around 48 to 72 hours, depend on the samples. And after incubation, we're gonna add MTT solution into each well, and then incubate around 4 hours for the activity of dehydrogenase. After the purple appear of the MTT formazine, we're gonna dissolve using DMSO, dissolve the formazine using DMSO, and then incubate in that for 2 hours. And then we measure the purple color normally at 570, I'm sorry, it is 570 nanometer using the spectrophotometer. Oops. And we often test at different concentration of the samples to make a graph like this, make a line like this. Here in my study, I also used this is A375 and A2058. These are melanoma cells. You know melanoma, right? That the skin cancer cells. And together with the skin cancer cells, I also test on the WS1 and NHDF cells. These are normal cells. They are fibroblasts, to compare the activity of the compound of the extract to the cancers and the normal cells. So I test at different extract, also methanol, hexane, chloroform, ethylene acetate, water. And I can see that some extract can give a really good result. For example, the purple one, that is the ethylene acetate extract. Yes. And on 2 kinds of melanoma cells. But for the normal cells, the extract can give lower, really low activity. I don't know exactly because I do not test the mechanism on this, that how the extract can be more toxicity to the cancer cell compared to the normal cells, but it also happens in some other extracts. And to go further, how the compound extract can kill the cancer cells, I use the to test. I think that the extract can use the apoptosis to kill the cancer cells. And then I to test whether the killing follow the apoptosis or not. Maybe you know apoptosis. Apoptosis is the programmed cells death. Is the cells is dead by in purpose. For example, the cells too old, or the cells is affected by the virus or bacteria or something. The cell need to be dead. So that will go to the apoptosis. So there's a lot of there, including intrinsic and extrinsic pathway of apoptosis. There's a lot of signal relating to apoptosis. I don't want to go further into this. It just means that there's some protein into the signal pathway of the apoptosis. For example, BCL2, MCL, BCL, BAC, or some others. And then to go further, we need to test whether some protein relating to apoptosis will be increased or decreased. And to do that, we will use real-time PCR to test at the mRNA level and using Western blot to test at the protein level. Maybe you know that PCR, polymerase chain reaction, that is the procedure that we amplify the gene of the DNA. Yeah, we add a lot of things into the tube and then add into the PCR machine. So from one DNA, it will amplify into 2, and 2 into 4, 4 into 8, 8 into 16, and yeah, and then amplify the gene into a lot of gene. And one kind of PCR really popular to test the protein, the expression of the gene, that is the real-time PCR or qPCR. Real-time PCR also a kind of PCR, but we add the fluorescence. We use the fluorescent primer. So if the if there are more gene amplified, the signal of the fluorescence increase. So from that one, we can draw a graph. The machine will give us a graph, a line like this with increasing. If the graph increase, that means the increasing of the amplification of the gene. Yes, and from that graph, we can draw into a graph like this. For example, in here, I use different concentration of my extract, and I test at 2 groups, BAC, on my left side is the some gene relating to increase the apoptosis, and on my right side is BCL2, BCLXL, and CL1. That is some genes relating to decrease or inhibit apoptosis. So we can see that at mRNA level, my extract can increase some genes of triggering apoptosis, some gene increasing apoptosis, and also decrease, for example, BCL2. That is a gene that reduces the apoptosis. That means my sample can enhance the apoptosis of the cancer cells. And here I test on the melanoma cells. And final one is the Western blot. Maybe I go quite long. Okay, I will go shortly. You know that Western blot is the technique, really popular technique to test the protein expression, to test the signaling pathway. At first, we have the protein sample. We're gonna add onto the membrane. We use the antibody. First is the primary antibody to detect the protein, and then we use the secondary antibody that detect the primary antibody, and the secondary antibody linked to the HRB. This is the host radix peroxidase. an enzyme that will change the substrate into the signal, and then we will detect the signal to see whether our protein is fresh or not. Here is the method of the Western blot. At first, we run the SDS page on gel, load the protein onto the gel, and then we transfer into the membrane, and then we block with PSA or skim mill, and then we add the antibody, the first antibody, and then the second antibody, and then we add the substrate and the chemical to expose the band onto the film. Onto the film here, if you see the band thicker, that means the protein is fresh more. The more protein is fresh. So in here, I also measure those kind of protein relating to apoptosis and compare the thickness of the band. So I can range into two graphs. Onto my left side, also the protein relating to increasing apoptosis. On the right side, also the protein relating to reducing apoptosis. And similar with the real-time PCR result, my extract can increase some triggering protein relating to enhanced apoptosis and reducing some protein relating to reducing apoptosis. And that's all. I want to introduce some in vitro method to measure the antimicrobial, antioxidant, and anti-cancer activity, some basic training for the activity. And if you have some question or something you do not understand or some comment, please give me. Thank you.
Hello.
Hello.
I would like you to get me clear on these steps for the PCR. Like you said, you prepare the sample, like maybe the DNA or the RNA from the cancer cell. Then you design a primer.
Yes.
After designing the primer, you mix the sample. You mix the sample with the primer. I don't know whether I've got it clearly.
You don't know how to do that?
Yes, I just got that. The first step you prepare you prepare the extract from DNA or RNA from the cancer cell. Then you mix with the primer.
Yes, thank you. Thank you for your question. Normally, I extract the mRNA. And then I use reverse transcriptase into the DNA, cDNA, and then we apply into the real-time PCR. Yes, and then I design the primer to enhance the primer for the PCR after that DNA. And then we add the fluorescence. The fluorescence will interact with the dimer DNA. If the higher fluorescence, the higher fluorescence signal, that means the higher the DNA was amplified already. So from that signal, we can measure the amount of DNA from the beginning. So we can think that we can give the number of the DNA of the beginning of our sample. Is my answer satisfied or?
Yes. Yeah, I've got it. Then I wanted to supplement on the techniques that we use, the PCR techniques. You talked about the real-time PCR and the Western blot.
Yes.
Now, we were also taught that there is a reverse transcription PCR,
Oh yeah,
whereby it converts the NRA to complementary DNA using
yeah.
the reverse transcriptase.
Yes, yeah.
Also, we were also given the digital PCR, which provides the absolute quantification of the nucleic acid without the need for standard values. And also the multiplex PCR. Yeah,
Yeah, multiplex PCR. Yeah,
yeah.
yeah, really
Mm.
powerful. Okay, thank you for sharing. Okay, any students, you have any question?
I think to add on that,
Oh yes, it's the time for
I am still on the PCR. Uh, uh, I tried. Okay, during the time when they were teaching us, we used to compare the advantages of those
yeah,
of the anti-cancer assays, like the PCR. For it has high
yeah,
sensitivity. It can detect the low absorbance mutations, and
yes,
another
yeah.
advantage, it can also amplify the target sequences with a precise sequence.
Yes,
Though it also has some limitation, like it requires expertise. That one in designing primer and optimizing condition that can be challenging, and also it is expensive. Yeah.
yeah, yeah. Expensive. I see. So,
Okay, unfortunately, because of the time limitation, I have to end this discussion session. But I would like to say thank you very much for Dr. Han for a very insightful presentation. As we come to the end of this lecture, I will return this session to the MC.
Hello.
okay. Hello.
I first of all, I would like to thank our professor for the wonderful presentation, and I would like you to share with us the presentation.
Send you the file, right?
Yes, the file.
Okay, okay. So, okay, I will send the file. Okay, maybe I will read to Dr. Hiền and then she will she will send you. Okay.
All right. Thank you.
Okay. Okay, thank you so much for listening and give some um, comment for me. Thank you, everybody.
Thank you, Ms. Calista and our honorable speaker, Dr. Hang Bigdo, for the insightful session. Now, we would like to call upon Professor Padlina Chanisa Putri to present a token of appreciation for our wonderful moderator and honorable speaker. To the documentation team, the floor is yours.
Okay, thank you, MC. Thank you very much, Doctor, for your interesting presentation. This is our appreciation to you for your insightful talk on exploring the potential activity of natural product through in vitro bioactivity studies. Your expertise and passion for this field have truly inspired us and expanded our understanding of this important area of research. Thank you for sharing your knowledge and contributing to our program this morning. Thank you. Okay, thank you for your sharing, Doctor.
Oh, can I say something? Now I can see my certificate. Thank you so
Okay.
much for the certificate.
Welcome.
Yes, you prepare really carefully. Thank you.
Thank you.
So beautiful.
Once again, thank you, Ms. Calista and the Honorable Speaker, Dr. Hang Bikdo, and for the amazing session. Ladies and gentlemen, as we move on to Lecture 6, I invite you all to the next Zoom meeting session, which the link will be provided in the Zoom chat. See you all in Lecture 6, which will start very soon. Thank you.